Now, once a neurotransmitter has been released into the synaptic cleft, neurotransmitters obviously send chemical signals. So you don't want a chemical signal to be there for too long. So you have to, once you release the neurotransmitters, they have to do their work. They have to send a signal to the next cell down, and then they have to be released. They have to be removed from the area. So there are several ways they are removed. Some of the glial cells, GLIAL, those glial cells, will help to remove some neurotransmitters. That's one thing that happens. There are also some enzymes that break down neurotransmitters, that eat them up, that break them down. So, but one thing that happens is it's called breakdown of the neurotransmitter. So here is an example. One of the major neurotransmitters that we're going to learn about, it's called a bioamine. Bioamine just means a amino acid containing neurotransmitter. So one of the neurotransmitters we're going to learn about is called acetylcholine. Acetylcholine. It's made up of a combination of a fat and a neurotransmitter, or a section of an amino acid. So in between neurons, or at places where acetylcholine is transmitted, there is you're going to find the presence of an enzyme called acetylcholine esterase. So where you have neurons where acetylcholine is being transmitted, you're going to have the presence of an enzyme called acetylcholine esterase. So the word esterase or ase is usually what's used to refer to an enzyme in biology, acetylcholine esterase. It's usually the term that's used to refer to an enzyme. So you're going to have acetylcholine esterase breaking down acetylcholine. And what that does is it helps to remove the presence of the neurotransmitter from the synaptic cleft in order to allow the next signal to be transmitted. So it actually happens for effective, efficient signal transmission. Now, what will happen to some of that broken down material, like the acetate and the choline, which is left over from the acetylcholine that's broken down, it may be reused to make another neurotransmitter. So your body might break that down further, or it may reuse it to make more neurotransmitter. But certainly that is that's what happens during breakdown. So some neurotransmitters are broken down in the synaptic cleft or on the postsynaptic membrane. They're broken down, in this case, by enzymes. And we'll see why that's important later on when we look at different psychiatric medications and how they work. Now, there's also reuptake. Okay, this is another process by which neurotransmitters are removed from the synaptic cleft. They are taken back into the the terminal of the presynaptic cell. So they're going to be taken into the presynaptic terminal. That's called reuptake. So two very common ways that neurotransmitters are removed or taken away from the synaptic cleft are by reuptake and breakdown. In the case we just looked at of acetylcholine, breakdown happens in the presence of an enzyme called acetylcholine esterase. In some other neurotransmitter pathways, you're going to find reuptake, where the neurotransmitter is kind of hanging out there in the synaptic cleft, and then it's taken back up to be reused again. Okay, so that's breakdown and reuptake. Do you all get the difference? Okay, any questions? Okay, good.